Stuff from the previous that you got that I pushed out this dam. Alright. Let's see what we have. Okay. So let's go ahead and work backwards here. I'm gonna pull this out and go ahead and download this so I can do this here. That movement separately. Okay. All right. So, get rid of this. All right. So, and I'll adjust this after class or whatever. Because it's a PDF, I can't really edit it right now. But we'll go ahead and adjust this as we go. So anyway, I didn't get to eyes. We're going to cover eyes on the next exam. So that's the first thing we're going to do when we come back from Thanksgiving break, is after we review the graded test. We're going to talk about sensory systems and the evolution of eyes. The squid will yet again come back as our example of issues. Okay, so what we did do though is we talked about nervous system, and I think what we covered today here is this: what general pattern in the central nervous system can be described from sponges, Aeria, flatworms, segmented worms, arthropods, chordata, vertebrates? Kind of the same idea. You notice that sponges were added in there just because I want you to think about this. Sponges don't have them, right? So I probably wouldn't waste any time with that on the test. But you should be thinking about, kind of which organisms have what type of nervous system. And then this is basically what we covered today. Why can't octopus get smarter? And what limits invertebrate brain size? And the limitation there is that essentially the gut has to pass through their central nervous system. So there's a conflict between the gut development and brain development. Okay, so then if we go backwards, we talked about neuron function, and basically all of this was what we covered last time, which was basically neuron function and anyway, so there's going to be a picture of a neuron, and you'll have to label dendrites, cell body, axon, synapse, and I'll probably also have you identify what the sodium-potassium pump does. It's important that you note specifically the number, and I'll have that on the test. It says show the number and which direction do sodium and potassium get pumped in or out. And remember, it's 3 sodiums get pumped out and 2 potassiums get pumped in, and that's what establishes this negative voltage inside the cell, right? Then the sodium and potassium gated ion channels are basically how the sodium gets back in and the potassium gets out. So those are basically how the nerve action potential functions, is by letting sodium diffuse back in and potassium diffuse out. The cell does kind of like a polarization flip. It depolarizes, repolarizes quickly, and that's what nerve propagation is. Okay. Any questions? So what will happen there is I'll probably have a little graph, like the graph set up, but you will have to draw the little action potential graph, like I had here last time. I drew a little, resting voltage is at -70, and then it spikes up to almost +30, and then it drops back down to less than -70 for a hyperpolarization, and then it goes back to -70 as the resting voltage. So that's kind of like the cycle of an action potential. I'll also have a little table that describes each one of the steps, and I'll ask you to put them in sequence. Like assuming the resting voltage is number 1, what would happen at 2, 3, 4, and 5? Back to 1. So that's kind of that part of the test is identifying the neuron parts, showing how the action potential goes, and then listing the sequence, describing the sequence of things that happen. Right? Sound good? At the very end of it, I will probably ask you a little 2-point question of how do myelin sheaths alter nerve function? And we described last time that myelin sheaths basically cover up like 90% of the axon. And what that does is it lets the action potential jump down or skip down the axon. And so that jumping down the axon speeds up the nerve impulse by a significant amount. It makes it run 10 or more times faster than an unmyelinated neuron. And so that would be a real simple description, right? Sound good? So that's what we covered last Thursday. All right. Then, before that, we were talking about muscle and skeletal systems. So, we have what determines the contractile strength and resistance of muscle contraction, compare and contrast extensor and flexor muscles, and then how does an earthworm skeleton function, the hydroskeleton, and then arthropod skeleton is an exoskeleton, and then there will be certainly a question about the limits of an exoskeleton. And the basic idea is that because the muscles are contained within the skeleton, as the body gets larger, the muscles can never outpace the skeletal mass. Because as the organism gets bigger, even though they presumably get 10 times stronger for every unit of length that they actually get 100 times more massive for every unit of length. So what happens is the body mass outpaces the strength of the muscles. Because the muscles are contained within the skeleton, the muscles can never actually grow large enough to really support the organism. I had a very elaborate graph that's a double wide plot. There will be a little graph on the test for you to fill out. So make sure you have those notes. Make sure you kind of have the basic idea. I'd be looking for like 2 J-shaped curves, one steeper, and that would be the mass, and one kind of shallower, and that would be the strength, right? It's kind of the idea. Because strength has to do with the cross-sectional area, the squared function, and mass has to do with volume, which is a cubical function. And if you were to plot x squared over x versus x cubed over x, that's kind of what the whole argument is. As the insect is bigger, the mass outpaces the strength. That's kind of the idea. And so that's why, for example, as far as insects go, like a Goliath beetle is about as big as it gets, about as big as your fist. And they're very slow. They're like almost like lumbering. So large arthropods are not very effective at moving around. Whereas vertebrates, because the muscles are on the outside of the skeleton, the muscle mass makes up more than the skeletal mass. The muscle mass, for example, in you, makes up, upwards of 60 or more % of your body mass. Your skeletal mass is less than 20% of your body mass. It would be like the opposite for an insect. their skeletal mass makes up over half their mass, and the muscle mass makes up less than half the mass. And so that works when you're really small, but when you're large, it doesn't work very well at all. So what you find is the exoskeleton works great if you're like less than a centimeter in size, but an endoskeleton works great if you're larger than a centimeter in size. So that's kind of like the break off there. All right, so there will be a question about compare and contrast hydroskeleton versus exoskeleton versus endoskeleton, and then why insects or arthropods never get to the same size as dinosaurs. And the basic idea there is that graph showing body size versus muscle strength versus mass. It's all a scaling thing. I know online there's also an argument that says the tracheal system can't support a large insect, but we're looking at all arthropods here. Arthropods never got the size of a blue whale, so even marine arthropods have been limited in size over evolutionary history. The arthropod design basically is not designed to be big, it's designed to be small. Okay, our favorite, small size. Okay, questions on that? All right, so we got some good stories there. Okay, and then we're going to have, if we go to the lecture before that, we talked about muscles in terms of the sliding filament theory. So muscles, I'll have a diagram that has kind of like the actin filaments and the myosin bundles and the Z lines, and I'll have you label the actin, the myosin heads, the Z lines, identify what the sarcomere is, and what is the direction, or how does a muscle contract in terms of the directionality. I'll have this all listed in the question, but it'll be just like I drew on the board. And if you missed any of that or forgot it, there's videos on Canvas of me blabbering about it over code, so you can watch it again if you want to. But so label the muscle sarcomere, basically, is what you're going to do. And then I'll have a sequence of events that occur for contraction of the muscle. And just assume that we're going to start that when the calcium is released by the motor neuron and releases tropomyosin from the actin. And then what is the next step? What happens next is kind of like the basic idea for muscle contraction. All right, so I'll have a little table that has a list of events, and you'll have to basically put them in order. If I start with number 1 as being calcium released, okay? So we'll be prepared for that. It's real simple. Just 1, 2, 3, 4, 5, 6. And then there will be a question at the end is what is rigor mortis? And specifically, at which stage does rigor mortis occur? And rigor mortis occurs when the myosin head is bound to the actin, right? That's when the myosin head is stuck on the actin after it's full. And what detaches the myosin head from the actin? ATP. So what rigor mortis is when you run out of ATP because you're no longer doing respiration or circulation, the myosin heads stick to the actin and they can't ever move. That's what rigor mortis is. So just make sure you kind of remember that, because I'll ask you at which stage above in the table does rigor mortis happen, and it'll be the stage where the myosin head is stuck to the actin filament after. Okay. I'll probably have a real simple question. I'll have a compare and contrast section again, and in this one I'll probably have a little compare and contrast skeletal muscle versus smooth muscle. So there were a couple features of the two, namely organization, number of mitochondria, which one is innervated by nervous system versus hormone or physical contraction. but just go ahead and, be prepared to do a little simple smooth muscle versus striated muscle comparison. All right? Then, I think, moving backwards, we had I came back from our last test, and this was stuff that I pasted over from, the previous exam. That is nitrogenous waste removal in arthropods. The big issue with arthropods, you should remember this, is they don't have ciliated cells. So arthropods don't use nephridia to do water balance or get rid of nitrogenous waste. They use essentially ion pumps in some sort of a organ structure in order to regulate water balance. So the crayfish and shrimp use a green gland, which is basically a water pump. So when they're living in salt water, they're using it to retain water and excrete salts. When they're living in fresh water, they're using it to excrete water and retain salts. So the green gland is kind of like this osmotic pump that they use at their antennae to regulate water balance. They just simply get rid of ammonia through their gills. When they move on to land, though, you can't just get rid of ammonia, you have to convert it to something else. So we went through this whole how the Malpighian tubule works. Malpighian tubules are found in spiders, myriapods, and insects. So it seems to be kind of a common solution for arthropods. Anyway, the Malpighian tubules had a very specific, I'll have a diagram that shows kind of an insect gut, and you'll have to kind of identify what's going on. And the main theme with the Malpighian tubules was the Malpighian tubules are ion pumps. They pump salt ions from the hemocoel into the gut, and what happens is water and soluble uric acid follow into the gut. And probably somewhere you should indicate that the hemocoel is around pH 7. The uric acid is soluble, but when the uric acid gets to the hindgut, it's around a pH 5 or so, and the uric acid precipitates through the crystal. And that's how they separate uric acid from their water and salt content. And then they're able to reclaim the water in their hindgut once the uric acid is separated from it. And you should be able to identify why or what the name of the waste that an insect excretes. And it's called frass. The frass basically is a mixture of feces and solid uric acid. Okay? Any questions on that? Again, I blah 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 for like almost an hour on this, so I can't redo that for you right now. But you can go back and look at your notes, and I believe that you can find that on this video as well. All right, and then I went through this whole thing about respiration onto land. Arthropods probably colonized land. Well, they're the first animals to ever be on land. Probably myriapods or chelicerates, some sort of scorpion or some sort of a millipede probably was the original land animal. And then later, insects evolved onto land from probably a crustacean ancestor. So we have, at least 3, probably 4 different evolution events of arthropods colonizing land before vertebrates or mollusks ever did. And they kind of did it in different ways. So, so the big question there is, what's the big deal about living on land? And other than there being a open salad bar that no one else is using, which is a big incentive, there's also a lot more oxygen available on land, right? About 32 times more oxygen available in the atmosphere than there is in land, in the water. And it doesn't depend on temperature. In the water, if the water is cold, there's more oxygen. If it's warm, there's less oxygen. So, so the benefits of living on land is it's not temperature dependent. There's always the same amount of oxygen, and it's way more than water. Other benefit is the ratio of oxygen to CO2 is on land it's like 500 to 1 ratio, and in the water it's like 4 to 1. So it's like way easier to exchange oxygen versus carbon dioxide in the atmosphere than it is in the water. A big problem, of course, of living in the atmosphere is desiccation, drying out, and that's kind of the issue. So you're going to have to basically show how the major group of arthropods have colonized land. Chelicerates with their hook lungs, crustaceans with their water bubble over their gill, the little bugs do that. And then the insects basically have developed this tracheal system that carries the atmospheric oxygen all the way to the cells without using blood or hemolymph at all. And then I'll have a little question about the diving beetle and how the diving beetle uses a physical gill. Is basically, I went through the whole story on that one, but the general notion is that the bubble is being supported by the nitrogen, which makes up 78% of the atmospheric gas. And then as the insect consumes the oxygen and makes CO2, the CO2 readily dissolves out of the bubble, and to replace it, oxygen comes out of the water because it has low solubility in the water. And so the bubble basically acts like a gill, even though it's basically a physical feature. So a brief description of how the diving beetle does respiration would be cool. And that's all basically an aquatic organism using atmospheric respiration underwater. I think that's pretty complete. I don't think I really have to change this study guide. Is there anything? Oh yeah, the only thing I have to change, and I will do this, is at the very bottom, we're not going to talk about any sensory cells or eyes. Okay, that's the part I have to change. Sound good? Okay, so let's take a long 10-minute break, like quarter after here, and I need to go get my sharks. And make sure you get pictures of this because I'm going to race as soon as I get back. And then we're going to do the sharks and bony fish, or the cartilaginous fish and the bony fish today. So the main thing though is the shark dissection is kind of messy. So I'm going to recommend, for one, that I wouldn't have any food items anywhere near this kind of they're black all, they're kind of like slimy and fishy. So I wouldn't.
That's a good idea.
And then two, is you probably want to have like any valuable items or clothing things kind of. So they don't smell too bad, but you don't really want to go to your next class. So long 10-minute break, and then we'll do that. And it's not too bad. I think it's pretty straightforward. Okay.